Hello, today we'll be studying how our living organisms on earth are made up of, from bacteria to giant trees to humans. Every organism is made up of cells. But here is a twist. Not all cells built are the same. Some are simple while others are complex and organized. Today we are exploring one of the most fundamental concepts in biology, prokaryotic versus eukaryotic cells. What makes them different and why they are important, and how did these tiny cellular differences shape the evolution of life on earth. Let's dive in. So cells are mainly classified in two major categories, that are prokaryotic and eukaryotic cells. So the word prokaryote means before, pro means before, karyon means nucleus, which ultimately means a cell with no true membrane bound nucleus, under which examples are bacteria and archaea. Similarly, when we talk about eukaryotic cells, eu, which means true, karyon again means nucleus, which ultimately means a cell with true membrane bound nucleus. Example of these kind of cells are animal and plant cells. Uh, these cells also differ in their size. Eukaryotic cells are more forward when compared to prokaryotic cells in size, where prokaryotic cells are only 1 to 5 micrometer and eukaryotic cell on the other hand are 10 to 100 micrometer large. These cells have something in common. These things in common. One of them is plasma membrane, which is the outer boundary of the cell, which is made up of lipid. This regulates exchange of oxygen, nutrients and waste. Also maintain surface to volume ratio. The next thing is cytosol, which is the intracellular fluid present in the cell. This harbors cellular components, organelles and carry out metabolic functions. The third thing which is common is DNA, which is deoxyribonucleic acid, the genetic material of all the cells. The last thing is ribosomes, which is made up of rRNA, which is ribosomal RNA, and proteins. These help in synthesizing proteins from the DNA. Now let's dive deeper into each type of cells. In prokaryotic cell, these are simplest and oldest form of cells on the earth, as we saw example being bacteria and archaea. Their DNA is not enclosed by a membrane and it is present in the cytoplasm free floating. These cells are small, simple and highly efficient. The single cell can function efficiently even without complex cellular organelles. They have outer structures such as capsule, which have gelatinous outer layer and protects from desiccation and phagocytosis, which helps in getting adhered to the surface. Inside to which is a cell wall, which is made up of peptidoglycan layer, and it is outside the plasma membrane, which provides support and protection from the osmotic pressure. Apart from these, there is a pili, which are hair-like protein appendages and help in transfer of genetic material to other organisms. The next is flagella, which is made up of flagellin protein and helps in anchoring to the basal body and gives a mobility to the bacteria or prokaryotic cells. Now we'll see what their inner components are. First, their genetic material, which is free floating as discussed, is known as nucleoid. These are non-membranous and made up of non-membranous DNA molecule and help in containing genetic information. Next is plasmid. These are circular double-stranded DNA which is present apart from their genetic information, and these gives antibiotic resistance to the bacteria. Also there is 70s ribosomes which are free floating in the cytosol, helps in formation of protein. These 70s ribosomes are made up of two subunits, that is 50s which is the largest subunit and 30s which is the smaller subunit, and helps in translation of the protein from the DNA. Now on the other hand, we will look the complex system which is the eukaryotic cell. These are large and much more organized, and their nucleus is true nucleus, and they have membrane bound organelles and work with proper coordination. So as unlike prokaryotic cell, eukaryotic cell many organelles like nucleus, membrane bound nucleus, mitochondria, endoplasmic reticulum, and vesicles, lysosome, Golgi apparatus, and their functions are well coordinated among themselves. So the first and foremost is the control center, which is the nucleus. So the nucleus also have nucleolus and ATS ribosomes. Nucleus is double membraned, which have outer and inner membrane with pores, and it contain DNA in the form of chromatin, which helps in controlling all the cellular activities. Nucleolus is a non-membranous core inside the nucleus. It helps in the production of rRNA and assembling the ribosome. The ribosome here is ATS, apart from unlike prokaryote which has 70s. It have larger subunit of 60s and smaller subunit of 40s. It may be found free or attached to the endoplasmic reticulum and help in translating proteins from DNA via mRNA. Now this is the factory for the production of protein. Once the mRNA is formed, it goes through this pathway. So from nucleus, attached is rough endoplasmic reticulum, which is of two types, rough endoplasmic reticulum and smooth endoplasmic reticulum. Rough endoplasmic reticulum have a network which are the network of cisternae and have ribosomes present on them, making it rough, and helps in the production of secretory and glycoproteins. Smooth endoplasmic reticulum on the other hand are without ribosomes, so these are smooth in nature and helps in synthesis of lipid, detoxify drugs, and stores calcium. Now Golgi apparatus, which are flattened stacks, receives from cis phase and modifies packages lipid and carbohydrates and shifts via transport vessel to the trans phase. So once the protein is formed, it passes through endoplasmic reticulum and received by Golgi complex for further modifications to transport it to their designated place. So apart from these major organelles, there are other organelles such as mitochondria, lysosome, peroxisome. So major function of mitochondria is it helps in the formation of ATP via cellular respiration, and structure is also double membraned with inner cristae have matrix, inner cristae and matrix. Lysosome is also membrane bound, containing acidic hydrolytic enzymes which help in digesting molecules, phagocytosis and autophagy. Peroxisome is a single membrane which contain oxidative enzymes. It helps in detoxification of substrate by converting them into hydrogen peroxides and breaking them into water and oxygen. As we saw, eukaryotic cells are more complex and coordinated. So how these cellular framework work? So there are two components, cytoskeleton and centrosome. Cytoskeleton are the dynamic network of microtubules, microfilaments and intermediate filaments which give mechanical support, shape and motility to the cell. Centrosome on the other hand is specific to animal cell, which are pair of perpendicular centrioles in a microtubule matrix. it organizes these microspindles for the separation of chromosomes so that the cell division occurs in a coordinated way. Now plant cells. There are some exclusive organelles present in plant cells, such as plastids, which are chloroplast have stacked thylakoids in stroma for photosynthesis. These plastids are of two types, chloroplast and amyloplast. Amyloplast on the other hand are colorless and store starch. The second one is vacuoles. These are massive vesicles, helps in the storage of water and toxic waste, maintaining the water balance. The other one is cell wall. Cell wall is a rigid outer layer of cellulose microfibril. It helps in maintaining shape and control water intake by resisting osmotic pressure. So if we summarize these things in comparing prokaryote versus eukaryote, in case of size, prokaryote is small, having 1 to 5 micrometer size, and eukaryote is 10 to 100 micrometer size. Nucleus on the other hand is absent, but DNA is present in the form of nucleoid. In eukaryote, there is true nucleus is present with double membrane. Ribosome in prokaryote is 70S, which is divided into 50S, whose subunits are 50S and 30S. Eukaryote on the other hand have ribosomes of 80S and its subunits are 60S and 40S. DNA in prokaryote is present in circular free floating format, and in case of eukaryote it is linear and it is organized in the form of chromatin. Unique feature of prokaryote is presence of plasmids and peptidoglycan layer, and in case of eukaryote it is maintaining the endomembrane system, which we saw with endoplasmic reticulum, Golgi complex and other. Also it have mitochondria, and in case of plant cell a specific cellulose layer is present out of the cell as a cell wall, which is very specific to plant cell. Thank you for this, and inside the cell is many things are there to be explored, so stay curious.